This is part 2 of lecture 9. You should not be watching this unless you have finished watching the first part. You can find the link on eCentennial. So by now we have already labeled the nephrons, the different parts of it, and we learned about the relationship between the nephron and the blood vessels. And just a quick recap, there are different parts to the nephrons. Here is the Bowman's capsule, which is responsible for capturing the fluid that will be leaking out of the glomerulus. We then take the capture fluid, which is by now known as the filtrate. That will eventually become the urine, but along the path in the nephron, we will have the opportunity to modify the contents of the filtrate, right? So anything you want to take back from the filtrate, it's going to go back into the peritubular capillaries, and anything you want to add to it, you will be able to do so as well. So again, the nephron is the functional unit of the kidneys, right? It's important to know that this is not a single cell, right? It's not like where neuron is a single cell, right? Nephron is a tube that's made up of many cells, and in this picture, you can see the nephron dips into the renal medulla, right, and then it comes back out, and you have the blood vessels that are going around it at all times. So the renal corpuscle, you might remember, is basically the glomerulus, right? Glomerulus, which is the capillaries, plus the Bowman's capsule, right, which surrounds the entire glomerulus. So over here in this picture, you can see the yellow circle, the yellow balls here are the glomerulus, and they look like a ball of yarn, right? I told you that that's basically what this word means in Latin, I think. And you cannot see the Bowman's capsule here, right? Because in order for you to see the glomerulus, you have to dissolve away the Bowman's capsule, right? The Bowman's capsule essentially surrounds the entire glomerulus, right? So in this situation, you can think of the glomerulus as like the seed of a peach, for example, and the Bowman's capsule is everything that surrounds it. So you must take away the Bowman's capsule before you can see the glomerulus. So in the process of urine formation, there are essentially three major steps, and they don't necessarily occur one after the other. Some of these steps, like the second one and the third one, kind of occur simultaneously along the entire length of the along some parts of the nephron. But typically speaking, we have the glomerular filtration, tubular reabsorption, and tubular secretion. So as an analogy, you can think of cleaning a closet. Okay? So when I clean my closet, I typically would take out a bunch of stuff right out of the closet and put it on the floor. So that is a relatively non-selective process, right? So that's equivalent to glomerular filtration. Okay, you take a bunch of stuff out of your blood, some of which are actually valuable resources that you want to keep, right? But the initial step is to just kind of get a bunch of these things out of the way, out of the closet, right? And then you kind of sort through the pile later on. And so tubular reabsorption, it's like going through the pile that you've taken out and then seeing which piece of clothing you want to take back, and you're going to put it back into the closet. And as you put the stuff back into the closet, you might realize you missed like a sweater that you wanted to donate or get rid of, right? Then you would take it from the closet and put it back into the pile. And that's equivalent to the tubular secretion, right? So along the way, you might realize there is still some waste product in the blood that you wanted to get rid of, and so you will put it into the nephron. So typically speaking, right, the glomerular filtration involves taking stuff from the glomerulus, right, which the inside would be blood, and you will be taking things from the glomerulus and putting it into the Bowman's capsule, right? The Bowman's capsule, right? So once it goes into Bowman's capsule, right, it's now called the filtrate. Whereas tubular reabsorption is going to happen in the proximal tubule. It's going to happen in the loop of Henle. It's going to happen at the distal tubule and collecting duct. And what you're going to be doing is you're going to be taking things from inside of these structures, which is going to be the filtrate, and you will reabsorb them. You will take them back into the blood. And the blood vessels surrounding these structures, if you remember from previous slides, it's called the peritubular capillaries, right? So it's going to go back to your blood, right? This is a selective process. You don't obviously take back everything, right? That would defeat the purpose. You take back some of the things that you want, but just because of the way things are being transported, you might accidentally take back some unwanted material, which is okay, because that's what tubular secretion is for, right? Tubular secretion is going to happen also in some of these structures. So it's going to happen at the proximal tubule, right? Distal tubule and collecting duct. And it's going to be added to the filtrate. So actually, you will take things from the peritubular capillaries, right, which is blood, and then anything you missed in the previous steps, you're gonna put it back into the filtrate. Okay, so sorry that was a little bit confusing, but let me just rephrase it for you. The tubular reabsorption is to take back things that you've accidentally lost in step 1, and you're putting it back into the blood. Tubular secretion is to take some of the stuff in the blood that you might have missed, and then you're going to put it back into the filtrate. Okay, so as these two processes kind of happen along the ways of the nephron, you're going to be able to adjust the content of your urine. So let's quickly talk about what happens at the filtration steps. Okay, so glomerular filtration occurs when the blood goes into the glomerulus through the afferent arteriole. Remember, this is your afferent arteriole, and then it's going to become the glomerulus, which then is going to come out as the efferent arteriole. So the blood pressure in the glomerulus is actually quite high, 60 to 90 mmHg, compared to the standard 25 mmHg in most capillaries. So since it's capillaries, it's going to be leaky. Compound that with the high blood pressure, it's going to squeeze out a lot of stuff. So all these stuff that are squeezed out of the glomerulus is going to be captured by the Bowman's capsule. So not everything comes out, okay? Only small things that are dissolved in the plasma is going to be able to come out from the glomerulus, right? So large molecules like proteins, right, they are not going to be able to come out. Blood cells, right, your RBC, WBC, platelets, they are not going to be able to come out, right? Which makes sense, okay? Typically, we don't have red cells in our urine, right? If your urine has blood in it, that's of course a cause for concern. So these things typically don't get filtered out in a normal individual. They would just kind of continue on their way in the efferent arteriole. So anything that's not filtered will just continue to travel to the efferent arteriole, which then continues to the peritubular capillary, right? It's okay if you miss some waste product here, right? Remember, later on in the nephron, you will still have the opportunity to do tubular secretion, right? To take what you've missed, right, in this step from the blood and put it in the filtrate, which then will again eventually become the urine. So in this chart, you can see more clearly what is being filtered out at the renal corpuscle. So On the left here, this is the filtered components, filtered blood components, right, that is going into Bowman's capsule. So we have things like water, right, nitrogenous waste, small things like glucose, amino acids, vitamins, which are things that we actually want to keep. But remember, this first step is a non-selective process that is rather done without much concern as to what gets out, right? It's again like cleaning out the closet, right? You're just taking out a bunch of stuff and sorting through it later on. We have some salt, NaCl, HCO3, and then these are other waste products that we actually do want to get rid of. On the right here, these are things that are large. They're not going to go through, right? So we have the blood cells, we have the plasma proteins, and actually, water is gonna be here as well, right? We're not gonna end up having like a dry paste that goes into the efferent arteriole, right? Not all the water that goes in are going to be filtered, right? If that's true, then we're going to be dehydrated, fairly quickly, right? So, only maybe 60% of the blood that goes in, it's going to actually get filtered. Everything else will continue onward to the efferent arteriole. So, if you look at this list here, right, that essentially is everything that will not be filtered. And if you look at this list over here, that is basically what we call a protein-free plasma. You might remember from a long time ago, lecture 2, I think, I told you when we spin the blood down, you are going to have the hematocrit at the bottom, and then at the top here you're going to have the plasma, right? And in the plasma you have a bunch of stuff that are dissolved, right? Nutrient waste product as well as a protein. So if you take the plasma and you subtract the protein, that's essentially this list right here, right? So a lot of textbook, they call the glomerular filtrate, right, the stuff that comes out of the glomerulus, right, a protein-free plasma. Approximately same concentration, right, as you will find them in the plasma as well. So now this is the proximal tubule. Okay, the main function of proximal tubule is reabsorption. All right. Over here, you are going to take back a lot of the stuff that you've just lost in the glomerular filtration step. Right? There are a lot of valuable nutrients that you want to take back, such as water, right? Glucose, amino acid, all these nutrients and ions that you need will be reabsorbed through active and passive transport. Now here's the thing, every substance requires some sort of carrier, some sort of protein to take them back. And when all the carriers are working at full speed, at maximum capacity, then you won't be able to take them back anymore, right? We call this a threshold level for that substance. All right, the best way to visualize this is, imagine like a conveyor belt that's moving, okay, and then there is some workers here, right? The workers represent the carriers, and this conveyor belt is basically the stuff in the filtrate, and you're trying to pick out these big dots, okay? So there are these big dots that you have to take out, and there are the small dots, okay? So if all the workers are working as fast as possible, right, they won't be able to pick up all the big red dots if there are a lot of them, right? So what I'm saying is, if you're trying to take back glucose, for example, right? So typically you can take back all the glucose that you want at the proximal tubule. However, if it's someone who has diabetes, for example, and then their blood glucose is really high, right? So these big red dots would be like the extra glucose, and all the workers, all the carriers that are supposed to be taking back the glucose are working at full speed already. Then you won't be able to take all the glucose back, and those glucose will continue on to the rest of the nephron, right? Which is why diabetic patients, if they're not on medication, they could have glucose present in their urine, right? Because you're not taking all of it back. So different substance is going to require different carriers, and each substance would therefore have a different threshold level, right? Which means the maximum amount that you can potentially take back. And if you don't have a carrier for a particular substance, then it won't get reabsorbed, right? So an example of that would be caffeine. If you drink caffeine, then the caffeine will not be reabsorbed at the proximal tubule because it's not a naturally occurring substance in the body. So we don't have a carrier designed to reabsorb caffeine, and that has implication, which we'll get into later on. So after the filtrate passed through the proximal tubule, 60 to 70% of what you've lost in the glomerular filtration step will now be taken back into the blood. And how much you reabsorb is going to be tightly regulated by hormones as well, right? Sometimes we don't need as much of a particular ion. Let's say you already load up on calcium, right? Then maybe you don't need to take as much back, right, from your filtrate, and you can just pee them out. Other times, if you are low in potassium, for example, right, then you might want to take more back at the proximal tubule. So the exact amount is going to be usually dependent on how much is already in the blood, and it's regulated by hormone. pH is also controlled here at the proximal tubule, right, the pH of the blood, and that is controlled by secretion of H plus and reabsorption of bicarbonate. Okay, so for example, let's say your blood is too acidic. Okay, blood is too acidic, and there could be various causes of this, which we're not going to go into. But if your blood is too acidic, that means you have too much H plus in blood, right? So you can secrete the H plus. Secrete the H plus means you're going to take this H plus from the blood and you're going to put it in the filtrate. Okay, so your urine is going to be a little bit more acidic. Okay, and at the same time you can reabsorb bicarbonate. So by taking the bicarbonate back into the blood, right, the bicarbonate can combine with the extra H plus that is making your blood acidic, and you will generate carbonic acid, for example. Okay, and this might be transported to the lungs where you can breathe it out as carbon dioxide. Right, so the kidney plays a role in controlling the blood pH. So after the proximal tubule, the filtrate will go into the loop of Henle. Right, so to explain the loop of Henle, I'm gonna have to draw a note for you. So feel free to draw along as we do in class, and hopefully this will make sense to you. So here we have the proximal tubule. Okay, proximal tubule, and we are going to have the descending limb here. We are going to have the ascending limb over here. Okay, so this is descending limb of loop of Henle, ascending limb of loop of Henle, and you probably remember, right, that the loop of Henle actually goes from the renal cortex, right, dip into the renal medulla, and then it comes back up later, right? So, at the proximal tubule, you will reabsorb 60 to 70% of the filtrate, right? And, depending on how much ions you reabsorbed, that's going to affect the concentration of the filtrate at this point. So, I'm just going to give a random number here, right? Let's say the concentration, the osmolarity at this point is 400, okay? Now, in the previous a video I told you when we were labeling the kidneys, right? I told you the renal cortex has lower osmolarity compared to the renal medulla, which has higher osmolarity, right? And that's actually important for the process of urine formation. Okay, so I'm just gonna make up some numbers here, right? I'm gonna say the osmolarity of the kidney, right, which is what this stuff is. This is the kidney, right, the outside. This is the nephron. So we're gonna say this is 600 unit, 800, 1000, 12,000. Okay, so we are going to see what happens when the filtrate goes down the descending limb. Okay. Now, the descending limb, right, on the way down, it is only permeable the walls. The walls of the nephron is only permeable to HCl. Okay, so let me put that down for you. Descending limb, right, only permeable to H2O. That means only water can move, okay, in or out, right? Let's skip to a new page for a moment, okay, and explain this a little bit more for you first, okay? So let's say I have two chambers. You know, we kind of talked about this before in the course, but you know, just to refresh your memory. Let's say I have 400 unit osmolarity here compared to 600 unit osmolarity here, and this membrane only allows H2O to pass, right? So my question to you in class was, you know, which way will water go, right? Will it go from the lower osmolarity to higher osmolarity, or the other way around? Okay, so the correct answer is water will always go from the lower concentration to the high concentration, so that it will be able to balance out the salt content, right? And you know, I remember I'm telling you in class, if you put a spoon of salt in your mouth, for example, then you can feel the saliva sucking out of your mouth, right? So this is essentially the extra salt, right, sucking the water out of the 400 unit here. So you know, keeping this in mind, let's go back to our drawing, right? When the 400 unit filtrate comes down the descending limb, and compare that to the 600 unit outside, what's going to happen is the water from the filtrate will be reabsorbed. Okay, so H2O. So the water will briefly come out into the kidneys tissue, and then right away it will go into the peritubular capillaries. Okay, so I'm not going to draw that capillaries because it's going to be confusing to the diagram, but you remember the peritubular capillary is always next to your nephron, right? So it's going to come out a little bit, and then it's going to go directly into the blood, and that's a good thing because we don't want to dilute our kidneys, right, too much, right? So after the filtrate comes down, you're going to reabsorb some water, which means your filtrate will now be more concentrated than before, right? So this is going to be 600, okay? And then when you go down further, now the kidney tissue is more concentrated than the filtrate, right? It's 800 now. So comparing 600 to 800, that will allow you to absorb even more water, right? And that's why we need the osmolarity of the kidney to be increasingly more concentrated as we go down into the medulla. We don't just want to absorb water at the initial step, right? We want to be able to absorb it all the way through, so that you know, you can take back as much water as possible to conserve them, right? So you're not going to become dehydrated. So after you take back some more water, it might become 800, and comparing 800 to 1000, you know, you can be able to absorb even more water, and finally at the bottom, right, it will become the same as the surrounding, right? So excuse me, the main function of the descending limb of loop of Henle is absorption of water, okay? Absorption of water, right? And more importantly, as you go down the loop of Henle, filtrate concentration increases. Okay, so if I use color to represent the concentration of the filtrate, right? So up here is kind of like a lighter yellow, right? And then as you go down, it's going to become a more intense yellow, and at the very bottom, right, it's going to be the most concentrated stuff. Okay, so as you go down the descending limb, the filtrate is going to get more concentrated. right? So then you're gonna go up the loop of Henle now. Okay, so on this side, we call this the ascending limb. Right, so ascending limb is a little bit different than the descending limb. Okay, so on the way up, right, NaCl are absorbed by active transport. Okay, so water cannot move, but you can take NaCl back through active transport, right? So the surrounding tissue, right, 1200 rather, 1800, 600, right? So what happens is when the filtrate goes up, right, you will actively take NaCl out of it, right? So you're gonna take some NaCl out, right? Take some NaCl out. So now the number is going to go down because you're removing the salt, right? So this is going to go back down to say 1000, right, to 800, and then to 600, right? So as you go up, you're going to be able to take back more NaCl, right? So the main function of the ascending limb here, right, is absorption of NaCl, right? And because you're taking the salt away, as you go up, the concentration of filtrate will go down, right? So now, again, I'll use the color to represent the concentration, right? Going back up is going to become lighter in color, okay? Lighter in color, okay? So the final concentration, it's hard to say, whether it's going to be the same or different than when it came from the proximal tubule. The key thing to remember here is, on the way down, you're going to be absorbing water. On the way up, you're going to be absorbing NaCl. Okay, and some people ask, is the water going to dilute the kidney? No, for two reasons. Number one, you're going to be just moving the water out of here and staying there transiently, right? It will immediately go back to the blood vessels. And number two, over here you're reabsorbing the water, but over here you're reabsorbing the NaCl, right? So overall they kind of cancel each other and maintain the osmolarity of the kidney tissue. Okay, so just quickly go back to the PowerPoint here. You can see the description for a loop of Henle. It's pretty much what I told you. You can read that on your own. And here it shows the relationship between the peritubular capillary and the loop of Henle. The peritubular capillary is really big, right? So the part that is just around the loop of Henle, they call that the vasa recta. Not very important for us, for our purpose. It's more important that you remember which part of the loop does what. Okay, so absorption of water on the way down, absorption of NaCl on the way up. So by the time the filtrate passed through the loop of Henle, you would have absorbed roughly 80 to 85% of everything you've initially lost at the glomerulus filtration. Okay, so you would absorb 60 to 70% at the proximal tubule, and then in the loop of Henle you would absorb an additional 10-15%. So by now you've pretty much taken back everything that you've wanted to keep, right? So the final adjustment of the urine content is going to happen at the distal tubule and collecting duct, and typically these locations are impermeable, right, to most solutes, right? And you're only going to be able to take things back through hormonal regulations. Okay, and we're going to look at a few hormones that plays a role in changing the permeability of the distal tubule and collecting duct, and therefore affecting the kind of substance that you're going to be able to reabsorb. So now we will talk about the regulatory function of the kidneys, and one of the ways that the kidney maintain homeostasis is through the regulation of blood pressure. So there is a system called a RAS system, which involves bringing blood pressure down when it is too high. So I'll be explaining this on in a separate video. And once you've watched that, then you can come back and read these slide yourself, because everything on the slides is going to be explained on the video. Okay, so I'm gonna stop here. You should go to part 3 to see the ROS system.